Thank you for that. But let's start. So the disease that we work with in our group, and that is the focus of my research, is called X-linked dystonia-parkinsonism. This is a very rare disorder that is an adult-onset disease and is characterized by symptoms of dystonia and then later on Parkinsonism. And one important aspect is that it mostly affects individuals of the island of Panay in the Philippines and mostly affects males. You will see why in a few seconds. The disease causes neuronal loss in the striatum and the thickening of the prefrontal cortex. So the very interesting aspect of this disease, that's why I am studying it, is because it's caused by a polymorphic transposable element insertion that is inserted in the 32nd intron of the TAF1 gene. TAF1 is a very important gene for the transcriptional regulation, so you can understand that its malfunction, it can lead to very bad consequences. So you see here on this graph that I show you the genome of a healthy individual, that there is no insertion, and then in these XDP patients, there is this transposon inserted. But what are really these transposable elements? So transposons, they are mobile genetic sequences in our genome that can still mobilize. Some of them, they have a very strong gene regulatory potential, and most importantly, they are at least 50% of the human genome. That is very surprising and interesting if you compare it to the only 2% protein coding genes, and many of the studies are focusing only on that 2%. So for me, it's very exciting that this huge amount of information is out there and a lot of things to discover. But then, because transposons, they can still mobilize in our genome, this mostly happens in the germline. And when that happens, then they can spread, these polymorphic insertions can spread in our population, and then that is a major disease risk in the human population. That's, for example, the case of XDP, this disease that we study. So the thing with transposons, since you can probably by now understand that it's very important to control them so they don't jump around in our genome, they are usually silenced by DNA methylation and H3K9 trimethylation. And here I show you how, in the schematics, how this complex works. So basically, there are these Krabs finger proteins that are transposon specific. They bind to the transposon, and then they recruit H3K9 trimethylation and DNA methylation to repress the transposon. And then the specific transposon that we study in this XDP disease is called SINE-VNTR-Alu or SVA insertion. So these transposons are of specific interest because they are primate-specific. Some of them are even human-specific. So they have 6 different families in total, and the E and the F families are human-specific. So that means that they are not in any other model organisms like mice or rats or pigs or anything. They are only present in humans. Interesting fact is that 1 out of every 60 human births has a new transposable element jumping, a new SVA. So I don't know how many we are in this room, but I guess at least one of us has unique insertions that only jumped in our germline, or in the parent's germline. So here you see the structure of an SVA insertion that is also called a repeat of repeats, and it's because it's composed of a hexameric repeat sequence, an Alu-like repeat sequence, a variable number of tandem repeats, and also a SINE-R repeat sequence that is coming from another transposon. So when you look at it, you can probably appreciate that it's very repetitive, and they are all over our genome, so it's very hard to study them. But despite that, we know that they are especially strong gene regulators in the brain, so that's especially interesting. And because of this repetitive nature, we have many, many challenges when we study these elements, when we use short-read sequencing. That was the consensus before. So the first main challenge is the mappability issues, that of course we don't know where the sequences come from because they are short and these insertions are very similar. The second one is this insertional polymorphisms. For example, in this disease, that it could be that you look at the transposon, but you don't know if it's in the reference genome or it's a new insertion. And the third one is that these transposons can have internal polymorphisms, so some mutations within them, and that makes it even more difficult to know which specific insertion you are looking at. So that's why nanopore sequencing came very, very handy for us, and it really helps us to overcome these challenges, because now we can actually map the transposons, we can annotate the polymorphic insertions, we can identify polymorphisms, and then we can also look at the DNA methylation that is very significant in case of these insertions. So now we actually can start to look at these elements in the human brain and see what effects they have, of course focusing on X-linked dystonia-parkinsonism, this disease that I introduced to you. So based on what I said, we hypothesized that in this XDP-SVA, it's covered by repressive epigenetic marks, and the presence of those marks affect the molecular phenotype of the disease and the disease onset. So we have two main research questions that I'm going to tell you about today. The first one is, what is the epigenetic status of this XDP-SVA? And the second one is, how does this affect the expression of the TUF1 gene? So first of all, to study this, we developed an XDP patient-derived model where we have iPSC cells that we converted to NPCs, neural progenitor cells, and we used that to study the epigenetic pattern of this insertion. We have these cell lines from healthy individuals and XDP patients. First, of course, we did nanopore sequencing because we wanted to see that this insertion is covered in DNA methylation or not. And here in this graph, you can see every line is a nanopore read, and the purple is the healthy individual, and the greenish color is the patient. And you see that the XDP-SVA is fully methylated, that is represented with the black dots in this graph. So then after that, we wanted to see if this insertion also has epigenetic marks like H3K9 trimethylation that are also known to repress transposons. So we did another approach called cut-and-run sequencing that is very similar to ChIP-seq, where you can see where your specific antibody binds, basically, your factor. And because transposons are repetitive, we decided, instead of using a bioinformatical approach that is very difficult, we designed qPCRs, where we have one primer inside, one outside of the transposon, and then just use the libraries to do the qPCR. And we do that. When we do that, we see that this transposon is highly enriched in this epigenetic mark in the genome. So now we have established that this XDP-SVA is covered by DNA methylation and H3K9 trimethylation in the genome of the patients. Next, of course, we wanted to see what happens to the gene expression at this locus. So we did RNA sequencing. And as you can see here in the last three lines that are patient lines, you see that where the transposon is inserted, the intron is retained. So this was a very interesting observation that was also observed in other groups. But I want to highlight here that the level of gene expression is very similar among cell lines. And I will get back to this a bit later. Then next, what we wanted to see what happens to this intron retention when we remove the epigenetic marks around the transposon. So we removed them one by one and all of them together. And here you can see that basically when you remove DNA methylation, this pathogenic intron retention is way higher. So that means that the presence of the methylation protects the genome from the effect of this transposon. And then, of course, everything I showed you now was in a cell model. So now the main question is, how does this actually look in the brain of the patients? And then for that, we started another project that is actually quite recent, where we have prefrontal cortex postmortem tissues from healthy individuals and XDP patients, and then did very deep bulk RNA sequencing. The first interesting thing that we discovered is that both in healthy individuals and XDP patients, young SVAs, so the ones that are only present in humans, they are lower methylated compared to old SVAs that are also present in primates, in other primates. Then when we looked at this XDP-SVA locus, here you see 9 different individuals, and you can see, so the violin plots, they show the CpG methylation of the XDP-SVA as a whole. And you can see that the level of methylation is very different. So some samples are very, very lowly methylated at this locus, but not others. And then next, of course, we wanted to see how does the gene expression look at this locus in the brain of the patients. And surprisingly, we found that there is intron retention, but the level of intron retention was very different in different individuals. So now if you can remember the plot I showed you about the cell lines, there we saw the same level of intron retention, basically. But from this data, it looks like that the picture in the human brain, as expected, is much more complicated than in a cell culture. So I think that's also a very important take-home message, that things are more complicated in the actual brain than in the dish. And then now, of course, we are working on trying to find the correlation between the presence of DNA methylation and the level of TUF1 misregulation. And it's an interesting aspect because since XDP is an adult-onset disease, so people are born with this mutation, but they only get sick when they get around 40 years old. So now we have the hypothesis that for some reason DNA methylation is lower over that region, and that's when the TUF1 gene expression gets worse, and that's when the patients get the symptoms. And then with that, I just would like to summarize what I told you today. So I showed you that this XDP-SVA causes intron retention, that it's covered in DNA methylation and H3K9 trimethylation, and that the presence of especially DNA methylation affects the molecular disease phenotype. DNA methylation and TAF1 alterations vary amongst patients, and it highlights very much the importance of using adequate models when studying brain disorders and adequate methods such as long-read sequencing. And then, of course, XDP-SVA could be a potential target for therapeutic approaches in the future, because we could imagine a scenario where we can, for example, by using CRISPR approaches, we could reinstate DNA methylation in the loci and hopefully get back TAF1 expression levels to normal. And with that, I would like to thank for the Jacobson Lab, because this was a great team effort, our funding agencies, and for you for listening to me. Thank you.